Hi, Assalamualaikum, and a very good day to Dr. Ani and my fellow friends. My name is Nurul Aiman AFIQAH binti Nur Azruddin, and today I will be explaining about characteristics of animal, which is the first subtopic of the topic animal evolution and diversity. So let's get into it. But before that, I would like to introduce to all of you my group members, and from the left is me. And then we have KHAIRUNNISA binti Roslan, Sarah AMANI binti Muhammad Razali, and lastly Muhammad AQIL bin AZHAM. So firstly, as you can see, this is a map. And it is a map of the world. And as you can see, there are emoticons of animals all around the world. So basically, it is just saying that animals existed all around the world. In this subtopic, I will be explaining about animals are multicellular, heterotrophs, motile, and they reproduce sexually and asexually. So the first characteristic of animals is that animals are multicellular. As you can see at the left picture is the picture of multicellular cell, and then animal also lacks cell wall. As you can see that the outer cell of animal is cell membrane, while the plant cell have cell wall as their outer cell. So that's the differences between animal cell and plant cell. Next, animals are heterotrophs. As you can see, the bird is eating something and the bear is eating a fish. So, in order for an animal to get the acquired nutrients and nutrients for the body, they have to consume other sources. So, these sources are the fish and the thing that this bird is eating. So, that is basically what heterotrophs are. They have to consume other sources in order to get nutrients that they need. So, the next characteristic is animals are motile. So what is motile? Motile is the ability of plants, organisms, and very small forms of life to be able to move by themselves. So as in the picture, the cheetah is running and it is representing the animals are motile. In addition, muscle and nerve tissues are also both characteristics of animal. They allow motility and a variety of flexible movements. Next, coordinated movements allow animals to seek for their mates and seek shelter and a suitable climate. These behaviors that have allowed animals to live in all habitats and to become vastly diverse. Lastly is animals' reproduction. We know that most of the animals reproduce sexually. And as you can see in the left picture, it is the picture of cells dividing until they become a zygote. So in the right picture, it's a picture showing a process of fertilization for certain animals that go through sexual reproduction. So next, this is the example of animal that reproduce asexually, like starfish. We know that animals are descended from a single common ancestor, thus forming a single lineage on the tree of life. Within the animal lineage are two main branches, which are the vertebrates and invertebrates. So the left picture is the picture where animal with backbones, which is also known as the vertebrates animal. And then this is the picture, the right picture is showing the animal without backbone, which is known as invertebrates. They move by a specific structure in their body, like liquid for earthworm, because they do not have any backbone. That's why they are known as invertebrates. So the last part for this subtopic is I'm going to explain the colonial flagellate hypothesis. So the colonial flagellate hypothesis claims that the first animals evolved from flagellated protists that lived in colonies. As you can see the first picture right here, it is a motile flagellates from an aggregate, and then the colony of cells from a hollow sphere. We know that these flagellated protists live in colonies, right? And then according to this hypothesis, as colonial cells become more and more interdependent, some of the cells specialize to perform different tasks, and the cell of the colony become differentiated. So as in the third picture, the specialization of cells occurs for reproduction. And then the enfolding of this colony of cell creates tissues. As cells become ever more specialized and differentiated, true animals evolved as cells from tissues and then organs. So basically, what this hypothesis is trying to tell us is that the first animal ever evolved is from the flagellated protists. That's all for me. Thank you. I will be passing the next topic to my other group members. Thank you for listening.
Assalamualaikum warahmatullahi wabarakatuh. Today I am going to present about ancestry of animals. In this topic, I will be talking about colonial flagellate hypothesis. The hypothesis states that animals are descended from an ancestor that resemble a hollow spherical colony of flagellated cells. Flagellate means organism with one or more flagella. This is a colony of cells with flagella. It is thought that animals come from these colonies of cells. Next, available evidence suggests they evolved from a proteus called choanoflagellates. Because they are most likely resemble the last single-cell ancestor of animals. They are the closest living relative of animals. This is a figure of choanoflagellates. This is a single unicellular, and this is a colony of choanoflagellates. This is how the colonial flagellate hypothesis occur. First, it comes from a unicellular flagellated proteus. Second, multiple flagellates make an aggregate. Aggregate means a whole formed by combining several separate elements. So this shows that several separate flagellates combine to make a whole colony. Third, the unspecialized flagellate cells form a hollow sphere because in the hypothesis states that a hollow spherical colony of flagellated cells. Fourth, specialized reproductive cells form, represented by the purple. The cells begin to specialize and is shown by this purple type of cells. It is specialized to perform a specific type of function which is a reproductive cell. Fifth, cells begin to fold in to make tissues. Based on the steps, first it begins with cell, second tissue, third organ. This is step to be. This shows that it will combine more to make a whole complete organism, which is animals. That is all. Thank you.
Hi everyone, I'm KHAIRUNNISA will continue to present about evolution of body plans. Animals have variety of body plans. For example, tiger have 4 legs, bird have 2 legs, and crab have 10 legs. All various animal body plans today were present by the Cambrian period around 500 million years ago. How such diversity have arisen in short period of geological time? As an animal develops, there are many possible outcomes regarding the number, position, size, and patterns of its body parts. Scientists now think that slight shifts in the DNA code and the expressions of genes called host genes are responsible for the major differences between animals that arise during development. For example, recent studies have shown that in insects, a mutation in a single host gene called UBX turns off the growth of some pairs of legs. Because of mutations in a single host gene millions of years ago, modern insects have fewer legs than modern crustaceans. A variant of the same host gene directs the development of the legs of both animals. So next, we will discuss about the phylogenetic tree of animals. Phylogenetic tree of animal is based on three things. First, morphological traits, living and fossil. Structural similarity that stems from having the common ancestor. For example, the similarity of the shape, size, and color. Second, the developmental homologies. The following picture are the examples of homology. The tail and the gill slits between fish and reptile, bird and human are homologous structures. They are different and have a different purpose, but they are similar and share common traits. Third, molecular traits, which is the RR sequences. Okay, so this is the phylogenetic tree of animal. This phylogenetic tree is a hypothesis and uses morphological and molecular data, which is rRNA sequencing, to determine which phyla are more closely related to one another. The two species are more related if they have more recent common ancestor. Okay, as I wrote A, B, and C here, between AB and BC, which one are more closely related? To know which one are more closely related, we would follow the line of both pair of species backward in the tree. Okay, I will draw the line between AB and BC. Okay, since A and B converge at a common ancestor first as we move backwards, and B only converge with C after its junction point with A, we can say that A and B are more related than B and C. Next, for instance, we can't say whether A and B are more closely related than C and D. Okay, I will say that this is D. We can't say whether A and B are closely related than C and D. That is because, by default, the horizontal axis of the tree doesn't represent the time in a direct way. So we can only compare the timing of branching events that occur in the same line edge. Okay, that's all for me. Thank you for listening.
Okay, next we move to types of symmetry. There are three types of symmetry in animal kingdom. First, asymmetry. Second, bilateral symmetry, and third is radial symmetry. For asymmetry animals have no particular patterns to body shape, and there are lack of symmetry. We can see this sponge, red-billed bird, and fiddler crab, which some part of their body are asymmetry with the other one. And next is radial symmetry. Radial symmetry can be seen by their bodies, which are organized circularly, similar like a wheel. And these organisms can be cut with any longitudinal cut through the central point to produce two identical halves. And some animals with radial symmetry can extend out in all directions from one center, such as jellyfish. These are some examples of radial symmetry, like Nidarius and Xenophorus animals, and Echinodon. Echinodon is like starfish. And next is bilateral symmetry. Bilateral symmetry is they have definite left and right half, and only a single longitudinal cut through the center line of the animal produce two equal halves. And this animal have defined anterior end and posterior end. This animal also have cephalization or localization of a brain and specialized sensory organs at the anterior end. Also, like human, we have cephalization, which is located at our brain. And the example of bilateral symmetry are like all animals except for asymmetry and radial symmetry animals. And next is embryological development, embryonic development. And the first tissue layers appear are called germ layers. Germ layers they give rise to the organs and organ system of complex animal. Animals such as cnidarians have only two tissue layers, which is ectoderm and endoderm, and these embryos are called diploblastic. This diploblastic animal only develop tissues but no specialized organs. And third is triploblastic, which have three tissue layers, which are ectoderm, endoderm, and mesoderm. Triploblastic animals can develop specialized organs. And next we know how triploblastics are categorized into two categories, which are protostomes, which mean proto which is first and stome is mouth, and deuterostome, which mean this term are used to determine what prior developed. For protostome, mouth developed first, and deuterostome is anus developed first and then mouth. There are stages of development in embryo. After fertilization, the cleavage occur, which is cell division without cell growth, and then blastula formation, and then xylem development. For first development is cleavage. Like we can see, the top view and side view of protostome are differently with deuterostomes, which is their spiral like that, and this is radial, which means radial symmetry. And this the daughter cells sit in groove, which is between, while the daughter cell of deuterostome is at the top. And for blastula formation, in protostomes, mouth appear at the blastopore, in here, mouth, and then in deuterostome, anus appear in blastospores, and then the mouth appear. Same goes to protostome, and then anus appear. The third development is xylem development. There are different between protostome and deuterostome. And for protostomes, mesoderm arises from cell located near the embryonic blastopore, and splitting occurs, and we can see the mesoderm will split to form a xylem. While deuterostomes, this is the mesoderm are linked with the gut, and next the mesoderm will enlarge at the outside, while the gut will meet in and fuse together. So the result will be the same, but the way they produce it are different. So that's all from us. Thank you.